Okay, so I'm Mallory, Caro. My project of research is Circulating Microfluidic Co-Culture Device for the Dynamic Analysis of the Tumor Secretome, and my advisors are Emmeline Miller and Dr. Melvin. So 1 in 8 American women, or approximately 12.5% of American women, will be diagnosed with breast cancer in their lifetime. And so on the chart to the right, we can see an estimated percentage of the different types of cancer cases in the United States in both males and females. And in American women, breast cancer takes up 30% of those new cancer cases, meaning that in 2020, approximately 273,879 of those new cancer cases in American women were breast cancer. And on the chart to the left, we can see the estimated mortality rate of different types of cancer in both males and females. And as observed in the chart, breast cancer is the second leading cause of cancer-related deaths in American women. So tumors are heterogeneous environments that consist of multiple cells, those being cancer, stromal, and immune cells. And so pictured as a breast cancer tumor, and in the tumor, these cells are constantly and continuously communicating with one another via paracrine signaling. So paracrine signaling is a type of cell-to-cell communication in which the cells that are near each other communicate through some sort of chemical messenger. And so looking at the picture, we see the secretory cell secreting that chemical messenger to the adjacent cell, allowing for that cell-to-cell communication to take place. And so those cancer, stromal, and immune cells mentioned in the previous slide communicate with one another via paracrine signaling and drive cancer progression and drug resistance. And so some cases of breast cancer can worsen into what is known as metastatic breast cancer. And so metastasis occurs when something moves or spreads from its primary site to a secondary site. In this case, we are talking about breast cancer cells migrating from their primary source, the breast, to a secondary source. And so some secondary sources could be the brain, bones, liver, lungs, and many other places within the human body. So to fabricate a wafer, we use the photomask and expose it to UV light. I never had the chance to do wafer fabrication in the lab, but Emmeline went through the process of making a wafer when I was not in the lab. And so, however, I have been trained on how to make a usable device, so I'll explain how that is done. And so using the silicone wafer shown in the top right picture, a polydimethylsiloxane or PDMS mixture is poured over the wafer and placed on a hot plate to sit overnight. And once the mixture bakes overnight, the PDMS slab is then removed from the wafer, and left on the slab should be the design of that was on the wafer, and this is due to the process of soft lithography. And soft lithography is a method used when fabricating a device in which a replica or stamp of the device is left behind on the PDMS. And so once peeled off the wafer, the PDMS slab is then cut into the 3 separate devices, and next holes are punched in the ports of the device. And lastly, the PDMS is plasma bonded onto a glass slide, and the device is ready for use. And so in the top right picture, we see a wafer design with a coin to scale, and it makes 3 devices. And then at the top, and then the bottom right picture, we can see a fully fabricated device bound to a microscope slide in a petri dish. So the device shown below is the current fully functional for adherent cell culturing device geometry and workflow. And it consists of 3 parallel channels that are separated by an array of microchannels. And so this allows for the passive diffusion of biomolecules from our 2 stagnant outer channels with a continuous flow generated in a center channel by a peristaltic pump. And so the goal of this work is to develop a microfluidic device capable of simultaneous co-culture of 2 different cell types that allows for the collection of the excreted biomolecules that cancer cells use to communicate with other cells. And so in using microfluidics, we are taking these complex, expensive processes and making them into scale natural processes. So a peristaltic pump generates a continuous flow of liquids. And so the role of the peristaltic pump in this project is to generate a continuous flowing center for our device into which our 2 outer culture channels can then flow through. And so with an off-chip sampling port that is incorporated into the peristaltic tubing, it allows for a fast and easy sampling of those extracellular media in order to identify those excreted biomolecules for cell-to-cell communication to occur. And so circled in yellow, we see that there is a binder clip, and that is used to hold the tubing shut to allow for the sampling of media. And pictured below is a legacy device generated by Joey Bauhoff that has helped guide my work on COMSOL. However, this is not the device we have been using in the lab. This is just to guide my COMSOL work. So using COMSOL and online software, I was able to generate this new geometry. And in this geometry, I focused on moving from a hexagonal shape to a trapezoidal shape. However, there's still more that needs to be done to this geometry. And so I'm trying to move from what I've shown in the top picture to what is shown in the bottom picture on COMSOL. So you may ask why we need to change this device if it is so close to working. And that is because of problems that we have seen arise. And so the next step on COMSOL is to work towards making the channels taller and wider to allow for the gelation of the hydrogel, and also to flip that geometry to switch from that hexagonal shape to the trapezoidal shape. And so in allowing the hydrogel barrier to gel, it will act as a fence to keep the middle channel from flooding. And then with the flip in geometry, we anticipate that it will alleviate an issue we were having where bubbles were forming from surface tension. And so I circled the hydrogel barrier. And so that's where that will act as the fence for our device. And so while I was in the lab, I took part in a seeding trial where we injected only the 2 outer channels with fibronectin. And in doing this, we were able to observe that not only that the middle channel did not flood, but also that the cells consistently adhered to the glass over the allotted time and after the fibronectin coating occurred. So in conclusion, seeding the cells using the fibronectin protocol yielded positive results, and loading only the 2 side channels proved to be successful in minimizing flooding of the device. So my future plans for this project include finalizing the device geometry on COMSOL and generating a mesh, and to begin validating the array arrangement and transport phenomena. And I want to give a special thanks to JC Busboom, Joey Balhoff, Josh Clavin, and Margaret Moe for all of their help and efforts toward this project. I'm finished with my presentation. I can take questions now.
Thank you, Mallory. Dr. Pardue, we'll start with you.
Thanks so much. I appreciate the presentation. Very good. I got some questions about your geometry. Can you go to one of your geometry slides? First of all, tell me what COMSOL is.
So COMSOL is an online software where you can generate an online device and run simulations. And so basically on COMSOL, I was working to build this new device in hopes that we could alleviate the problems that we saw arise.
So part B of my question, trying to keep to one question, is why 20 micrometers and 30? You had another slide. What dictates the size of that smaller channel? Why do you decide 20 or 30? Why not 10 or 5 or
So
2?
we needed to make the parts taller and wider because with the I guess sizes that we had, there were a lot of problems, like the surface tension problems arose. And so with those hydrogel barriers, we're hoping that it will cause a good gradient. And so our circulating media flow can work and give us good results.
Thanks, Mallory.
All right, Dr. Flake.
Yeah, so I had a question on the co-culturing of two types of cells. Why are you doing this? Why do you need 2 different types?
So we need the 2 separate types because tumors are made up of heterogeneous microenvironments. And so it's not just cancer cells, it's many different types of cancer cells. And we also have immune cells and stromal cells. And so we're trying to see how that cell-to-cell communication works that drives the cancer progression in like the tumors.
All right, thank you.
All right, thank you, Dr. Flake. Boz?
So which part of your presentation were you most concerned about getting your message across? And can you show me how you tackled that challenge by going to the slide?
I'm actually on the slide. So I was mainly worried and wanted to tackle the part where I was changing the device because that's the part where I came in. And so getting to change this device, it was going to help our work. And so hopefully once I finish finalizing the device, we can get it up and running and see if it will actually work.